Everybody, and thank you for joining this webinar. As you have just heard, this meeting will be recorded and put online, especially the presentations from our both speakers. This webinar comes in the frame of a European project named Booster, which is called Boosting Drought Tolerance in Key Cereals in the Area of Climate Change. Let me talk a bit shortly about this project. Booster aims to improve drought tolerance in both maize and teff, while simultaneously exploring the potential for transferring species-specific drought-responsive features and by exploiting natural genetic variation to achieve drought-tolerant genotypes, and by developing biostimulants derived from living organisms. So a booster is harnessing the natural resources available to develop new varieties of drought-tolerant agricultural crops. And today we are going to talk mainly about one crop or plant, which is a South African grass named Eragrostis nindensis. And for this, we will have two speakers. One, and the first one will be Dr. Llewelyn van der Pas, and then we will have Bojana Banović. So our first speaker is, let me call him Lou, is a postdoctoral fellow in the Department of Molecular and Cell Biology at the University of Cape Town under supervision of Prof. Llewelyn. His research is focusing on plant stress biology with a particular emphasis on drought and desiccation responses in vegetative tissues. During his PhD, he has investigated vegetative desiccation tolerant South African grass Eragrostis nindensis, making some large-scale proteomic studies to uncover some mechanisms that allow these plants to survive. His work also explored why older tissues lose desiccation tolerant capacity. As the only known desiccation tolerant grass with a close crop relative, this Eragrostis represents a key species within the EU booster. As I said, we're looking for genetic diversity that can serve drought tolerance. So within Booster, Llewelyn has completed an extensive and nationwide in South Africa survey and collection of different accessions of nindensis, associated rhizobacteria, isolating and screening them for plant growth promoting potential. So with this, Lou, I would like to welcome you, and I give you the floor. Please share your slides, and we're looking forward to hear. Maybe a last housekeeping aspect. So we will have the two talks one after the other. So after Lou has finished, I will introduce Bojana as a second speaker. And by the end, we will take questions by everyone to open the floor for discussion. I would advise you to write your questions as they come into the chat, and so we will pick them up afterwards during the question Q&A session. Lou, the slides are on. The floor is yours. Thank you very much.
Right. Thank you, Mark. Hi, everyone. So my nickname is Lou, but if you want to try, my pronunciation is Llewelyn. So as Mark said, I'm a postdoc researcher in the lab of Professor Llewelyn. And my talk today is going to be primarily about introducing Eragrostis nindensis. I've titled my section here, Drying to Survive, so exploring desiccation tolerance in Eragrostis nindensis. So I first want to give a little bit of background in that life is very important for all of biology, and we can kind of consider life in the context of water based on this sort of scale. So most of what we understand happens within this range of a water scale, right? But of course, with everything, we've got two extremes. So when we expand this to crops, for instance, we can think of maize as sitting in the more dehydration sensitive range. Things like Eragrostis teff which has some drought tolerance, sits in this range of dehydration tolerance. But then we also have a bunch of organisms that sit in the desiccation tolerance realm or in the anhydrobiosis realm. So these would be things like your seeds, your tardigrades, and of course, resurrection plants. So resurrection plants form a big part of what we research in Jill Farron's lab. And so I want to talk a little bit about these plants. So resurrection plants, they predominantly occur in Southern Africa, but you can find them all over the world. I've put some pictures of some representatives over here, and we broadly classify them based on how they treat their photosynthetic machinery. So we have our homoiochlorophyllous plants, try saying that 10 times in a row, and these basically retain some form of their photosynthetic machinery. So good examples would be the woody Myothamnus flabellifolia and Craterostigma pumila. On the other hand, we have our poikiochlorophyllous plants. So these tend to dismantle their thylakoids. As they're drying, they kind of look like they've completely senesced, for example, here with Xerophyta schlechteri. But amongst them, they kind of have three main core characteristics. All of them are able to dry down to about 10% relative water content. Many of them can survive in this desiccated state for prolonged periods. And when you give them water, they rapidly resume metabolism. So what would that kind of like look like? So if we think about Eragrostis nindensis, the video over here will show it going through a natural drying and rehydration cycle. So nindensis is commonly referred to as a lovegrass. It is a C4 grass, so it undergoes C4 photosynthesis. It's predominant in Southern Africa. Obviously, it has vegetative desiccation tolerance, but what makes this particular plant unique is, as you'll see with the video that's busy playing now, only some of the tissues actually resurrect. And we tend to refer to these as the younger leaves. So these display desiccation tolerance, whereas the older leaves lose this ability. So it's given us a very nice model to study desiccation sensitivity and tolerance in one species. What obviously makes this additionally unique is that it is a close relative to the orphan crop Eragrostis teff. So we can actually underpin key mechanisms that drive desiccation tolerance and drought tolerance and find those key genes which we could then apply to crops like maize to make them a bit more drought tolerant. So what we do in the plant stress lab is we use what is referred to as a systems biology approach. So we don't just target one specific thing, but we look at the entire plant as a whole. So we look at aspects related to its physiology. We conduct extensive microscopy work, including transmission electron microscopy. And then we do a lot of multi-omic stuff. So we do a lot of transcriptomics, proteomics, metabolomics, genomics, lipidomics, metagenomics, name it, we've done them all, right? And from that, we can gain an understanding of what those key genes and pathways are, which we could then use for targeted biotechnology, right? So we could introduce genes from species like nindensis into maize to make them more drought tolerant. But that's just looking at the above ground. Below ground, we obviously have this consortium of microbes that have co-evolved with these species. So our systems biology approach there is we target things like metagenomics to understand what that composition is like. We then also do targeted isolation of microbes, and we characterize them for key properties that could either enhance plant growth and or enhance their drought tolerance. So in this stream, we then direct this towards our biostimulant application. So nindensis, as I said, is a very unique system in that we can use a lot of information from that to apply in various different things. But nindensis we've developed as a fully robust system. So things that we've studied with nindensis, not exclusively us, but globally, we have a high quality genome for this. We've looked at its chromatin architecture under drought and well-watered conditions. We have a full panel transcriptome, proteome, metabolome, looking at these changes along this this dehydration, desiccation, rehydration cycle to find what's happening at all these key moments. We are currently, which my colleague Bojana will talk about, looking at the cystrome. We've conducted lipidomics, we've conducted transmutation electron microscopy, and we've done physiology. Other than the other resurrection plants, Xerophytus lecteri, nindensis is probably the only other species that has this much information on all levels of organization. So from physiology right down to which metabolites are being produced under drought and under desiccation. So what does nindensis present for booster? So as Mark alluded to, nindensis is one of the species that is covered within the booster framework. So we have nindensis, we have teff, and we have maize. So part of what we did with nindensis is we conducted an extensive survey of natural populations of nindensis in Southern Africa. Well, not South Africa, but mostly South Africa. I've just highlighted some of the sample sites that we've established in South Africa. And you can see that they occupy vastly different landscapes. So this landscape over here is in the Northern Cape. It's very close to the desert in Namibia, and you can see it's very barren, it's very dry. There's almost nothing really growing there. But then you go up to the Limpopo province up here, you can see it's a lot more greener. There's a lot more vegetation over here. But nindensis predominantly occupies this side of South Africa. So what did we do? We collected plants from these sites. We collected the rhizosphere, so this would be the soil that's associated with the roots themselves. We isolated bacteria. We characterized these for certain plant growth promoting properties. So can they fix nitrogen? Can they solubilize phosphate? Can they produce plant hormones? And can they survive simulated desiccation? From this, we were able to get down to about 24 candidate isolates that kind of ticked all of our boxes. We sequenced those top performing strains, and these have now been tested for their efficacy on maize and teff. Not in South Africa, but the teff was tested in Ethiopia and the maize in Europe. So in this approach, we've used our systems biology system, funny, system biology system, to characterize microbes that could feed into the biostimulant stream of booster. But of course, us being us, we've also looked at how we can target certain genes within nindensis. So this is what my colleague Bojana will talk about a little bit more, but what we did was we conducted a controlled dehydration experiment, similar to the video that you see now. We sampled this at key strategic water content so that we could find genes that are switched on exclusively under drought, which species like maize and teff would not be able to switch on. And those candidate genes now become excellent targets for genetic improvement in both maize and teff. So from this, we conducted a complete RNA and MOA-seq, which my colleague will talk about a little bit more. And what we're hoping to gain from that is find those areas where we can improve both maize and teff. So with that, this is basically nindensis in a very, very quick summary. And I should still be well within time, so I will do my little thank you slide. If anyone has any questions pertaining to resurrection plants in general or nindensis specifically, you are more than welcome to drop me an email. I happen to enjoy talking about these resurrection plants. They're really, really amazing plants. And it's always quite, even though I've worked with these plants for many years, it's always a wonderful sight to see them go from this almost dead-like state to fully green within 2 days. So with that, I will stop sharing and happy to answer any questions that you might have.
Thank you, Lou. Thank you very much for this nice introduction to the world of desiccation crops, especially the one of Lovegrass. We will now have, as said before, the second talk, and we will take all questions afterwards. So for the two speakers, please, to the attendants, don't hesitate to drop already the questions in the chat. That would help Lou probably to prepare his answer and you not to forget that question. So we talk about the next talk here will be given by Dr. Bojana Banović. Bojana, please apologize if I don't say your name properly.
Don't worry, it's perfect.
So you're a postdoctoral researcher at CREA in Bergamo, Italy, investigating how grasses like Eragrostis, nindensis, and teff survive drought using mRNA-seq and MOA-seq methods alongside genomic comparisons with maize. Your work aims to uncover the molecular mechanism, so what is happening behind the scenes within those plants in these drought resilient grasses and explore the potential to transfer these traits to maize. So you earned your PhD in plant sciences at the University of Belgrade in Serbia, and your specialty is in plant stress responses and bioinformatics. And you're also active in teaching, mentorship, and science outreach events such as Researchers' Night and Plant Fascination. So, Bojana, we're very looking forward to hear from your talk, and the floor is all yours.
Thank you very much. I will now restart my share. Hello, everybody. I want to say thank you for this opportunity to share a presentation with you, our research exactly. And thanks, Lou, for a very nice talk and for beautiful introduction. So what I talk more about today is the molecular response of Eragrostis nindensis to drought and desiccation. Actually, at this moment, we only studied the drought part.
No,
And
it's not really, sorry, excuse me. It's not really in presentation mode, I think. Is that possible? Because we still see like, maybe in view, you can say full screen.
Just a sec. Actually, in view, it doesn't offer.
Just go on top.
Normal outline notes. I don't see full screen anywhere. I'm on a Linux laptop, so I'm not sure if it is.
I think maybe down right you have like a red cross that opens it probably full, or is that already then the full one?
Layout. Where did you say? Sorry.
Down right your screen you have like a kind of red. I don't know if that would.
Yes, but it doesn't work.
Yeah. Okay, then I think we'll be fine like this. I wouldn't like to delay you much more.
But Sarah has my presentation. If you think it is better to.
I think it should be okay. I think it will be fine as long as your text it looks like big enough, so we should be able to see.
Okay, let's just recheck.
It should work. It should work like this. Thank
you. But it's still not full screen.
No, we still see like your whole, also the slides at the left side and so.
This is really. No, nothing works. I tried the red cross. I tried everything.
Okay,
It makes sense.
And in slideshow, it doesn't. I think that's already better.
Let's try now. Is it better? No?
Yeah. Just keep. Yeah, continue like this. I think that's the best. Now it's there. Perfect.
Okay, because I need to click several options to find the proper one. Okay, sorry guys for delay. Now that we are through the technical issues, I will just speed up a bit. Okay, so we study molecular response to drought in Eragrostis nindensis. As Mark already said in Booster, we have a strategy how to boost drought tolerance in sensitive plants like teff and maize. Well, actually, teff is more drought resilient than maize, but it is more drought sensitive than nindensis. So there it is. And actually, the strategy is to study how Eragrostis nindensis respond to drought, to water deficit at molecular level, and then to how to say, extract this knowledge, which drought features these plants has, and to through genomic comparative studies to try to find orthologous drought features in teff in maize, which are different, like switched off or a little bit changed, and to switch them back on or to change them a little bit to boost drought tolerance in them. And for this purpose, we studied 4 time points during drought response. As already mentioned, we had fully hydrated plants as a control, of course. Then we had a drought stress, which was 16 days after water withholding. And after 18 days, the plants were rewatered and then sampled 48 and 72 hours afterwards to see how rehydratation happens and what happens during this rehydratation. And for these purposes, we combined 2 approaches. One is molecular approach of transcriptomics, which means that we extracted total RNA, make mRNA libraries and sequence them. And the other also responds to transcriptomic changes, but at the level of differential transcription factor binding through the cis regulatory elements of these genes that are drought responsive. And after that, we simply analyzed these results and compared the data between 3 grasses. And this task is currently ongoing, and we have already interesting results. So the main point is when we find these orthologous to try through some genome editing strategies to introduce them to teff and to maize. And then what happens in the lab? Okay, we take the grounded leaf that Lou provided. Actually, he provides leaves. I ground them. I extract total RNA. I check its quality and quantity, make mRNA libraries. And for these purposes, we wanted a slightly longer fragment than usual. We just follow recommendation from the manufacturer how to produce these longer fragments. And after that, we test these libraries for quality and quantity using tape station and sequence the libraries to obtain what genes were active during these time points that we sampled. Then our colleagues from another CREA center, Primeta and Jacopo, analyze these data using the Nextflow pipeline that Jacopo made, and using these tools that you can see on this slide, which means that FastQC was used, of course, for the analysis of quality and quantity of the sequences. Then they trimmed and removed adapters, mapped them using STAR to reference genome, and finally, using Salmon, they quantified it. At the end, using DSeq program in R, they just made comparison between different conditions, fully hydrated, drought stress, then fully hydrated rehydration time point 1, and fully hydrated rehydration time point 2, to see what changes during these time points at mRNA level. After that, we proceed with MOA-seq. Of course, the same type of samples is used for both experiments in order for them to be compatible and to, how to say, complete our picture, what is going on at molecular level. So MOA-seq stands for micrococcal nuclease defined system occupancy analysis assay. What does this mean? This means in practical that we take DNA. We cross-link it with proteins that are bound to it to fix these proteins for the moment. Then we use micrococcal nuclease to cut only naked DNA, but naked under, of course, situation that they are not bound to proteins at that moment, specific moment in time. And when we cut, we use micrococcal nuclease, which has exonuclease and endonuclease activity. So it can cut also single nucleotides from end, but also within the DNA strand. And in that way, we release fragments of bound DNA with proteins. And after that, we, of course, cleanse this. We decross-link the proteins, and we only keep DNA fragments, which then we use for preparing, again, library. But this is specific a bit because after this library preparation, we need to cut very small bands, which are around 195 base pairs, which are really mononucleosomal bands. We want, because when you run mRNA-seq, you have some zebra pattern, and you want the lowest band that is equivalent to one nucleosome. We cut it, and then we send it to sequencing, and after that, Primet and Jacopo analyze this data as well. Using, again, a very specific pipeline for these purposes. And after checking quality and quantity of data, they merge these short fragments together using tool ngmerge, map them onto nindensis genome using STAR, and using MAC-S3 tool, they really read MOA footprints, which are really parts of genome to which transcription factors were bound at this specific point of time that we analyzed. And of course, at the end, they compared between different conditions. What is the difference in these MOA footprints in fully hydrated, drought-stressed, rehydrated plants? And the results shown first for RNA-seq show that there is a considerable number of genes which has changed expression during drought, and that the drought really fully answers at RNA level in comparison to rehydratation states. But these genes that are active during the drought stress are not the same genes that are active during rehydratation. Well, not completely identical. Some very small portion of genes are shared between the states, but the rehydratation point has a completely different set of genes, which means that it is not simply a recovery from drought, but that this process is actually a distinctive process in the plant, which occurs after it regains watering. And after that, we analyzed the MOA data. And in average, we had a really good score that has 300 millions read per replicate, and 90 millions were uniquely mapped in each, which scored more than 350,000 MOA peaks. And we checked for location of these peaks, and it is as expected, because when you think about where these cis-regulatory elements are in the genome, they're usually in intergenic regions primarily, but also in intronic regions. And really, more than half of our reads were mapped in intergenic regions, and more than 90 also in intergenic and in intronic regions. And the numbers were highly similar, for instance, between drought and rehydration 0.1, while they were very low at rehydration 0.2. So this rehydration 0.1 that occurs at 48 hours after receiving water again is really active point where plant is trying to recover all its processes back to the, let's say, regular physiological level. And when we looked at these MOA footprints, we were also interested if they are close to genes and to which genes. And by close, I mean we looked 5 kilobases upstream and 1 kilobase downstream the genes. And we counted that 70%, let's say roughly, were close to genes in each state, which means that these are really included in not really included. We don't have a proof still, but that they're really potential candidates that can be included in the regulation of expression of these genes. And finally, just a quick look what these genes do at metabolic level in a cell, because we need to yet do really deep analysis to see more details. But from a bird eye perspective, we can already see that all metabolic processes are far more active during drought stress. They are rested a bit in rehydration point 1, and they really slow down at rehydration point 2. What means slow down? They are back to normal physiological level, and of course they're always active. But you can see these blue and red, let's say, stripes or little squares, which actually show which genes are up. These are blue, or down-regulated during specific processes. And the more intense color, that means the more intense process. So you can see how active really at cellular level this plant is during only drought stress. You can only imagine what happens during desiccation. So with this, I will thank you, and I'm open to questions.
Thank you very much, Bojana, for this very interesting and more specific talk, let's say, as the follow-up of the presentation from Lou. I think we can open the floor for questions. I would propose, if people, we are not so many, so I think it's very nice if people are willing to ask their question directly rather than in the chat. So I see one already from Sandy. Sandy, do you want to ask and maybe precise your question to Lou?
Hello, good to see
Hello.
you. So yeah, I was very interested to see the kind of microbial work that you're doing. So have you got to the stage where you're testing any of the microbes that you isolated from the soil? And how do you plan to do that as a mixed kind of microbial sort of synthetic community or single? And then also, long question, I know, but do you have any ideas how you might apply?
Sure. So to the best of my knowledge, because this is being done in other labs, we're primarily testing them as single strains as first to see which ones really have individual impacts on overall plant growth and their tolerance. And I think we have narrowed this down to a couple of candidates that have really shown promising results. The next step would be obviously to combine them into a synthetic community or a synthetic consortium. And we have people who kind of specialize in how to apply this. And the most effective way, which we've also seen in the lab, is actually just to coat seeds with it. Because then you have the microbes that are in direct contact with whatever crop you are testing. And I mean, we've done independent experiments where we've also seen that, you know, when we do coat seeds, it has a much greater impact than, you know, if you just apply it directly to the soil. So I think that's the kind of direction that we are kind of like channeling this project in.
Thanks, Matt. I work in sugarcane, so there are no seeds, which was why I was quite interested. But thank you.
Well, I mean, from some of the work that we've done in our lab, even though the effect is not as pronounced when you're applying it as a, let's say, as a irrigated treatment, it still has some positive benefits compared to, you know, your water-only controls. So when you compare the mode of application, yes, then there's big differences. But again, you're still applying something that has an overall positive effect.
Thank you, Sandy, for your question, and Lou for your nice explanation. Is there any other question in the audience? Well, if not, let's let them come up with some questions, but I might still have, because I didn't know anything about desiccation plants, let's say. But we hear a lot about drought tolerance. So can you explain me a bit what the difference is between desiccation tolerance and drought tolerance? Is that anything in common, or how does that work?
Sure. Technically, they're kind of the same thing, at least when you think about desiccation tolerance. Well, when we talk about vegetative desiccation tolerance, so things like our resurrection plants. So desiccation tolerance is a mechanism of basically tolerating water loss, whereas drought tolerance is a similar mechanism, but it does not go to the extent of being able to be desiccated. So there are a lot of shared similarities in how plants respond. It's the activation of certain protective mechanisms, key pathways that are being activated. But in essence, what distinguishes the two is that if we take teff and nindensis as an example, teff being drought tolerant, nindensis being desiccation tolerant, is teff can only handle water loss up to a certain point. Beyond that, it dies. And it's that particular range where we know that something can tolerate water loss but cannot sustain that water loss, that we find those key genes that make something more desiccation tolerance. But to answer the big overarching question, desiccation tolerance is just a mechanism of surviving water limitation conditions.
I would
It is
just
a
add something at
yeah,
the end
sorry.
when you finish. No, finish, please, please.
No. You can chip in.
Because at molecular level, one of distinctions is senescence or programmed cell death, which is prevented in desiccation plants. They keep it at bay, we can say it like that, and they resurrect literally after they get water. This is one of distinctions that I am aware about.
Well, maybe. Yeah, Jill,
There is
you
Jill.
raise your hand, please.
Hi everyone. I've been listening quietly because I love it when Lou does the talking. And thanks for the very nice overview of our work, Lou. So senescence is a massive thing, and my lab is chasing this not only in nindensis but in other resurrection plant species, because the reality is when Lou was saying those two leaves in nindensis, one survives, the other one is desiccation sensitive, so we can compare them. The reality is those leaves senesce. They are not, they actually do the exact same thing as the tolerant, the ones that will survive. They transcribe very, very similarly, but they senesce below a water content of 35% water content, that critical level that defines a resurrection plant. And I think the reality of that study and some of our other studies is, and Lou's PhD looked at that, is how do resurrection plants stop that senescence process? And knowing now what goes on in the senescence of Eragrostis nindensis, because I've just published this paper, can we apply that to teff? And I sit looking at Barkel Gurma here, who's a current PhD student, will be working as a postdoc in Booster next year to literally do some of this genome editing in teff based on the findings we have in nindensis, but also based on the beautiful work that you're doing, Bojana.
Thank you, Jill, for these precisions. Yeah. Actually, I'd like to follow up with Bojana on what you have been saying before, and the whole presentation. You have been showing mainly work on basically on transcription, right? I mean, also the MOA-seq, let's say, is basically catching the promoters of the potential genes that are transcribed. So it's basically two questions. One question is did you find like already in your first analysis like some nice correspondence, like with the RNA-seq, some genes that are activated or deactivated corresponding to those promoters in the MOA-seq? Is that working? And on the other side, I mean, you have been focusing mainly on RNA, on transcription. I'm wondering if in this process, in this desiccation and rehydration process, are these the first processes that are being activated, or are we more actually, would it be more at the protein level or at other levels? I mean, I'm asking this to you, Bojana, but I mean, Lou, please feel
Yes, we
free
yes, we can answer together, no worries.
Sure.
So I will just start with the transcriptome part, and then we will proceed with rehydration. Okay, with transcriptome part, we do see the differences in expected group of genes, but there are also some subtle changes which we are interested in and looking for. And with cis regulatory elements, we are for the first time looking at the potential regulators of expression of these genes, which are much more interesting to control differences through very specific genome editing. You change only one nucleotide to change binding property of this cis element, for instance, and you can switch on or switch off something that is relevant for drought response. So yes, we are seeing differences, and we already have potential targets. Actually, we are in the process. I'm currently running comparative genomics part, which is already giving us results that we can found these kind of elements that are orthologous, which is also important, that you have already natively this element in teff and in maize, which can be a bit changed and doing something that is more like to make them survive water loss a bit better than usually. And for the rehydration part, well, I can say that we see a very specific signature. So this is a distinct process that happens after plant receives water, and it activates immediately, but it is intertwined. It is like a question, what was before, chicken or the egg, because these processes are completely intertwined. And Lu can answer from the protein part. I will answer just another sentence for the transcriptome part, that we do observe huge differences between drought state and rehydration point 1, as well as between two rehydration points. So as time goes, plant settles down its processes which were activated during water shortage and comes back to normal physiological levels. And then I leave Lu to answer about protein part.
Yeah, sure. So I'm actually in the process of writing up the proteomic study right now, and it is a mess, right? It's not always easy to as you say figure out what was there before and what's actually really driving these things. But what we find is that at least at the protein level, that even at 48 hours post rehydration, um, the the plant is still in a I don't want to say like a stress state, but it still has proteins that are associated with that stress response. And whether or not those just persist during that rehydration period to make sure that, it's not this huge influx of water that's now suddenly causing a lot of damage, because that's also a very big component, right? So you have cells that have kind of like folded in on themselves in a very orchestrated manner, and now suddenly you're expanding this with a lot of water, right? So during that initial recovery period, you kind of still have to have protective mechanisms there to make sure that you're not damaging your cell membranes, your cell walls, your organelles, because during that process, especially with nindensis, is when the thylakoid membranes are being reassembled. So you want to make sure that you don't have a lot of damaging things happening while you are getting back to your normal state, if you will. Um, but what I found looking at the proteins is even after 1 week after we've rehydrated them, they are still having a signature that indicates some sort of preemptive stress response. It's almost as if they are anticipating another dehydration cycle. And in fact, we've seen this with other species that we've studied, where some plants have a memory of these events and they kind of anticipate. It's almost as if they're sitting out in the wild, just waiting for it to like not rain for a good couple of days.
Yes, there is plant memory, absolutely. And I think all the plants have it, but we didn't look at it. And there is also a direction in science which studies this kind of, let's call it plant memory. And
Yeah.
it is really intriguing.
It is, it is. And I think it's those key factors that really also, just to go back to Mark's previous question, I think it's those memory factors and the sort of like priming effect that also help separate the drought from the desiccation tolerant species. But yeah, so what we found is that rehydration is a very crucial step, and looking at what happens there can also provide good insights into what other targets should we be looking at. Because yes, most of the research focuses is on studying the tolerance to the stress, but what happens after that stress is gone? How do you recover from that stress? So yeah, that's what I'm gonna say. There's more questions.
Okay. Well, I mean, people are a bit shy today in the chat, but I mean, I've been really intrigued by both your presentations. So I might have some more questions. One thing one thing that I think is important within the Booster project, we talk about genomic diversity within nindensis and teff, and how to apply this to maize. I mean, how do you see that? Is it really something that is transferable? Can we really consider, transferring one gene or a cis element to some
Sorry, Mark,
concept?
something is with your mic. I can hear only one part, but not the other. Can you please repeat your question?
Oh, do you hear me?
Yes, now it's much better.
Oh, okay. I just moved the buds in the ears, so
Okay.
I don't know what that did. So my question was more about like how much of this information that you're accumulating, this knowledge on teff and mainly in nindensis, is then transferable, and how are we going to do that? Because I mean, for me, the question is really like we have genes. I mean, the genes are. I mean, maize is very far from nindensis, right? So how could that be applied in maize, and does that question or that approach really make sense?
Yes, okay. So I will start, and Lou, you can chip in whenever. So the point about this transferability is exactly comparative genomics, which I mentioned before. So we did the same experiment like this one in nindensis. We did it with teff, and we did it with maize, and always in drought stress, but that level of water loss that teff and maize can survive, of course. And we look at their transcriptome profiles and transcription factor binding profiles, and we compare. And we find similarities, we find differences. But with genomic comparison work, you search for orthology. So you search for the genes that came from the same ancestral gene. So when speciation happened, these species inherited this gene, which means that probably this gene has the same function in all the species. It is different than paralogous genes, which came through gene duplication event and can have different function later on. It's like hemoglobin is one example. Hemoglobin in men and in mice is example of orthology, while hemoglobin, looking at, for instance, different globin genes, it is a paralogous example. So through this orthology, we find the elements that are orthologous but different. So in nindensis, they are active, for instance, and in maize or in teff, not active or not so active, because there is also difference in quantity, in how much expression you get and what these express gene do. So this is the approach, and it is transferable very easily because now you have a CRISPR-Cas methodology that allows you to enter into genome to change single nucleotide that you are interested in, for instance, this cis-regulatory element that can make the difference for the gene expression that is controlled by this cis element. If I need to be more descriptive, just let me know. I am not sure if I'm being clear enough.
I think that's already very clear. And I don't see any other. Jill also applause
Thank
for
you.
your.
Thank
And there is no people popping up to ask additional questions, so I am supposing that this was very clear.
you. Great.
Maybe more directed to Lou in that case. Lou, you have been mentioning microbials, I mean microbes associated, but in a very dry context, and I wonder what the advantage or what really the added value of microbes. I don't know if they would be as endophytes or as just soil microbes. What would they bring in that process, and how would that work? Do you have any idea about that?
Yeah, so our current working hypothesis is that if we just consider the resurrection plants in some of the more drier areas, is that the microbes that are associated with them have co-evolved to handle that environment, right? So we could basically describe that there's this trade-off happening between the microbes and the resurrection plants. What we have seen from metagenomics is that there are definitely enrichments of certain types of phyla that are only found with these resurrection plants. Um, and our proposed mode of action is because they're so well adapted to very harsh environments where, water is scarce, they have to be in a desiccated state for prolonged periods of time, both the plants and the microbes is that when you transfer this to a species that is slightly less drought tolerant, like maize, that they would then exchange those same sort of properties with maize and thereby enhancing them. So some of the things that we look for specifically is whether or not these microbes are able to produce the plant hormone IAA. So if you have IAA being produced, you can have roots that are much bigger, more dense, so they can tap into deeper reservoirs of water. So that's one way that we can apply a microbe to enhance a basic drought tolerant mechanism. So if you can increase the root system, you can obviously enhance the water that it can access. So what we find is many of these microbes that we've screened for, they do seem to have this particular property over and above the fact that they can enhance other plant growth promoting properties like make more nitrogen available, make more phosphate available. But the basic premise lies on the fact that these microbes have become so well adapted to a very dry environment that if you put them in an environment where you challenge them with drought, they're already well adapted to that situation. And because they've adapted with plants that are also well adapted to that environment, there's this nice give and take that happens between these two systems. But of course, the big caveat is it's not one microbe that operates in isolation. It's the entire microbiome, right? But that's our kind of working hypothesis of why they're good to target.
Okay, thank you. I see that Tekle has a question here, so I might want to follow up on this. Tekle, please.
Yes, thank you. I saw the comparison was made between 3 crops and maize and the other 2 Eragrostis crop plants. So maize in our case grows in a highland area, so it gets enough moisture. So wouldn't the comparison would have been better with sorghum or like millets that are really stress tolerant, drought tolerant in our case? So why was maize chosen as a comparator to the 2 Eragrostis?
Well, I can start because maize is European most wanted crop at the moment, and it is significantly hit with the drought because yield of maize dropped a lot due to long droughts that Europe had, and this is why it was accepted as one of the main. But this strategy applies to all. So the strategy that was designed for booster can also be applied to sorghum as well. So it is really a beautiful approach for this kind of investigation that you are interested in.
Yeah, I think the comment of Tekle makes a lot of sense. If you look at the project as such, if it would have been designed for an African based context, we would definitely have chosen other crops. The Booster project, it's an EU project, which is and specifically this call was more oriented towards improvement of
Yes.
drought tolerance in Europe. With the knowledge and expertise from more dry region, which in that case was South Africa and Ethiopia. So yes, Tekle, you wanted to say something still?
No. Just somebody disturbed me. I'm in Nairobi, in a hotel.
Okay. I actually had maybe a last question before we go each one for lunch or for late afternoon in some cases. You're talking about nindensis, Lovegrass. So two questions actually. First, why do you call it Lovegrass? Second, why is it? No, it's not why. Is it only localized or is it endemic to South Africa or is it this Lovegrass that we find? Do we find it like everywhere on the planet in similar climates, or how is it spread globally? Thank you.
So the the lovegrass is one of the common names that we have for, well, in fact, all of the Eragrostis species, their common name is lovegrass. But for example, Eragrostis curvula, also widely grown in South Africa, is called weeping lovegrass, for example. So where it actually comes from is Eros is the Greek word for the Greek god of love, and grossis is I think the Greek word for grass, if I'm not mistaken. So ergo love grass, and it stems from the way that the spikelets, which is the grass name for the floral parts, it actually looks like a little bit like a heart. So if you actually want to have a good look, if you Google Eragrostis superba, it has a very beautiful spikelet that actually looks like little hearts that are stacked together. So that's where that comes from. I just like to use the word lovegrass because science can be a bit boring, and lovegrass is a bit more exciting. It also gets people excited. As for its distribution, I do stand corrected, but I think most Eragrostis are found in southern Africa. I think nindensis, spreads, it goes as far as up to, I think, mid-Africa, but I'm not sure about its global distribution. If there are different Eragrostis species globally, I just happen to know nindensis very well and its distribution. But I do know that it's one of the biggest grass families in South Africa. So a lot of the grass species that we have are from Eragrostis.
Okay. I see Romana raising her hand. Please, Romana, we're listening to you.
Hi, good afternoon, and
Good afternoon.
congratulations for two great talks. Now that I'm hearing about there's someone there studying sorghum, I remember, and also in terms of water tolerance and resistance to climate change. I'm currently based in Portugal, and we do a lot of citizen science activities, actually projects, to study also water drought-tolerant crops and alternative crops. One of them is sorghum. And I was wondering if you guys are also involved in these kinds of activities, and if the local community in South Africa is involved, maybe with the sampling of the lovegrass, or if it's something that have crossed your mind. Just a curiosity.
So we've done other work with not within the booster scope, but where we've looked at other crops like sorghum in particular. We also work with other orphan crops like Bambara. So we kind of spread ourselves to kind of target different crops that are very well suited to the African context. Because a big part of Jill's work is food security, particularly in Africa, targeting crops that are well adapted to Africa, but have not really been extensively researched. So Bambara, teff, sorghum. But as for your lovegrass question, it's not a grass that's particularly interesting to most South Africans. I think in South Africa we predominantly use teff for horse feeding. So I know quite a lot of the horse stables use teff to feed the horses. And curvula is predominantly used on farms for foraging. So things like your sheep and goats, they tend to eat that. But it's not one that most people are particularly interested in, except us, because we love nindensis. Actually, no, we don't. Nindensis is a horrible plant to work with. It never behaves the way you want it to behave. But deep down, we actually do love it.
Does any plant behave the way you want?
Nope.
I don't think so.
That's the beauty of it.
Yeah,
And the
yeah.
beauty of life. But I can chip in also for the part of citizenship science and stuff like that. In Serbia, I did a lot of this, but we did another resurrection plant, which is called Ramonda. And it cannot be sampled, for instance, because it is endemorelic species, which is preserved and cannot be used as such uncontrollably. So you can ask for permissions to go to specific landscape to take it. So you need also to think about these kind of things when you plan your work.
Nice. Thank you.
You're welcome.
Okay. I mean, I think we had a wonderful discussion, actually. We learned a lot about the work that Booster is doing, about your passion for Lovegrass, both of you, and why it is called Lovegrass. I think we have passed even the hour, so if there is no other question, I think we can leave it for now. Unless you still have, or if you want still to say a last few words, maybe Bojana, just if you want to say something.
I just want to thank you for this opportunity to present our work also in this kind of format. I prefer this kind of formats than to other ones because they're more relaxed. People can relax and chat a bit. So I really like this, and thank you very much.
Thank you.
Yeah, I just want to echo what Bojana said. It's always nice. One of the things is Jill and I can go on and on and on about how wonderful these resurrection plants are. It's just because they're just really, really fascinating plants, and it's always a huge pleasure for us to really talk about it. So again, thanks for organizing this, and thanks for giving us a small little platform to talk about these wonderful plants.
Thank you very much, both of you. Definitely was great. We heard great talks. People were participating and are clapping the hands. So I'm sure you have made impact today on people. And I see nice comments coming in the chat. With this, thank you very much. Have a nice day, weekend, end of year festivities. Looking forward to see you online or physically for one of the meetings. Bye-bye.
Thank you very much. You too. Bye-bye.
Bye, Mark.
Bye, Tekla.
Yeah, I see Bakal. Where is he from? Where is he from? Ethiopia.
Yeah, I don't know.
Yeah, he's from Ethiopia.
Okay.
I know, yeah, but I'm saying, are you still in Ethiopia or you studying elsewhere? Yeah.
I don't know.
Yeah, you just answered. Yes, it's okay.
Okay. Okay. Tekla.
Bye-bye.
Bye-bye.
Thank you. Thank you.